Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh and good day to everyone. Thank you and welcome to all the participants and judges of the second International Conference on Tropical Resources and Sustainable Sciences organized by University of Malaysia, Kelantan. I am Muhammad Shafiq bin Hamdan from Bioaromatic Research Center of Excellence, University of Malaysia, Pahang, going to present my research entitled Characterization of Equilaria malacensis Kell cells using SEM and somatic embryogenesis associated genes identification. These are my team, Ms. Aimi Wahidah, Dr. Siti Hajar Noor Syarani, which is the corresponding author of this publication, and Dr. Saiful Nizam Tajuddin. First of all, what is Equilaria malacensis? So, Equilaria malacensis is one of Malaysia's agawut or kaharu or depu or karas species that produce high-value resinous wood, and essential oil that are being used as incense, perfumery, and in traditional medicine. So this species was included in Appendix 2 of Convention on International Trade in Endangered Species or CITES, and it continues to decrease in nature due to illegal harvesting and high demand from industry. So recently our government agency like FRIM, Forest Research Institute, has established action plan to conserve this species by conducting artificial propagation, R&D, and enforcement to overcome this problem. So as you can see here, this is the natural agawut tree or Equilaria malacensis in forest. This is the harvested agawut from this tree, and this is an native people or honestly working on to extract the essential oil. However, they do not operate the factory anymore due to the problems of the resource. So nowadays people move into plantation to relocate the species, and currently we are focusing to multiply this tree for conservation purpose. Now I'm going to introduce you the term of somatic embryogenesis. It is an in vitro technology or tissue culture technology to generate embryogenic cells by somatic cells that are being in use and undergone morphological and biochemical changes. So we have reviewed some of the research that had been conducted a few years ago, and then we found that some of the tissue culture technology of for this particular species had been done for different purposes, such as for a regeneration system, for establishment of cell suspension culture, to study the functional genes, and then one research study tissue culture for free disease seedling, and another one for the genetic enhancement to produce more phytochemical. Generally, there are three stages in the somatic embryogenesis, which are initiation, proliferation, and maturation. So the advantage of this technology can produce the plantlets within short time, and it is possible by using the bioreactor to generate the somatic embryogenesis. So, as a quality control for this technology, some researchers already studied deep into the advanced technologies such as microscopic imaging, omics technology, and computational biology. So, for the microscopic imaging, some of them using the scanning electron microscope, and they found that uh, a morphological marker to verify the somatic embryogenesis by detection of this specific structure, which is extracellular matrix ECM or extracellular matrix surfaces network ECMSN. So this pictures taken from the sample from pine tree, and this one is from the brachypodium or barley tree. Apart from that, some of researchers going into the molecular aspect to identify uh, associated genes related to somatic embryogenesis for biomarker purpose and functional genomic study. So some of researchers already successfully isolate some of the related genes such as leafy cotyledon, shell, baby boom, and somatic embryogenesis receptor-like kinase gene. So these genes are involved in transcription for the somatic embryogenesis pathway in the molecular level. Next is the objective of this study. First, we want to optimize careless induction techniques by using scanning electron microscope, and the second objective is to identify somatic embryogenesis associated genes by using polymerase chain reaction analysis. For the materials and method, the first method is explant collection and sterilization. We harvest the sample from Andauropic forest. The sample was kept in liquid nitrogen during the sample collection, and then we sterilize the sample by using method by so this picture how we running the tap water. So some of the method using the Clorox as well as ethanol to remove the fungi from the surface of the explant. This is some of we use different explant in this experiment. So this is the nodal part of the plant. This is the seed, and this is the leaf, young leaf, to regenerate the calyx. Secondly, for somatic embryo induction, we use basal media contain MS salts and vitamins, as well as the right as the base media. Then we adjust the pH at 5.7 to 5.8 and autoclave them. So after autoclave, we pour the media onto the petri dish and let them solidify under laminar airflow cabinet. Then we pipetting 5 different hormone concentration and combination according to the protocol developed by 5 of group researchers from our references. Then we maintain the culture under dark or light condition with 25° plus minus 2 Celsius temperature in our growth chamber. For electron microscopy, so we didn't doing chemical fixation to the sample, just native sample, and then we put onto the specimen holder and then let the machine operate. Then we observe the samples using FI Quanta 450 scanning electron microscope with Everhard Thornley detector. So we pick the best pictures with the resolution from 10 to 500 times magnification. So next we doing the DNA extraction and PCR analysis. So the sample with 100 milligram from the candy that we were regenerate stem and leaf for the control had been pulverized in liquid nitrogen. Then we extract the genomic DNA using the kit which was FavorPrep, the implant genomic DNA extraction mini kit. So we follow their protocol, and then we check their quality by using nano-Q spectrophotometer, and then we run the polymerase chain reaction using the design primer for different genes that we already obtained from the database from this website, NCBI or National Center for Biotechnology Information. And then we run the PCR using thermocycler using specific annealing temperature. So these are the primer that we have designed by using the information from the database. There were 4 genes. Then these are the accession number from the sequence source from the database. So finally we isolate and sequence the gene fragments. Basically after the machine run amplify the genes, so we separate the PCR product on agarose gel, and then we purify and send the sample for sequencing. So after we got the result, we analyze manually using software, and then we compare the sequence to available sequence in gene bank also from the same uh, website, and then we compare the desired protein using the constraint based multiple alignment tool Cobalt that was also available from the NCBI homepage. So the result from our study, and this is table 2, and optimized parameters for somatic embryo induction. So there are different parameters that we tested during the experiments, and we found that the best explant to induce the somatic embryo was the young leaf. Then the sterilization method using optimized method were by running tap water for 15 minutes, using twin 20 for 1 minute, using 70% ethanol for 30 seconds, 5% chlorox for 2 minutes, and 0.2% benomyl for 2 minutes for fungi sterilization. And then the best media was MS media with the gyrite and sucrose. The best pH media was 5.7 to 5.8, and the most important was the plant growth regulators combination use. So we found BAP and NAA was the best PGR to induce somatic embryo under dark condition and 35 to 26 degrees Celsius incubation temperature. So this one just pictures different explant use that we use. This is the nodal part, a leaf, and seed of the Acrylium alkensis. So these are the pictures for callus formation in picture A. 30 days incubation, 50 days incubation, 3 months incubation, and 4 months incubation in dark condition. So this one is the embryogenic callus formation image after 5 months and 6 months incubation in dark. So these are the microscopic analysis using SEM. So the red arrow shows the part of ECM, which was the extracellular metric. So we compare this metric with the previous researchers' result. We found they have like a net And then the yellow arrow shows the isogeometric shape of the embryonic callus. So this is the result for PCR analysis. So these are the analytic temperature that we use to run the PCR with different DNA samples and different targeted genes. So here some of the result PCR product we identify the best result prior to sequencing. So however we found only SERP gene, somatic embryo receptor kinase genes producing the best band from the agarose gel. So we send for sequencing. These are the result from the sequencing. Unfortunately, the bioinformatic analysis result hit the ILS superfamily, so it's not the SERP genes. Finally, the conclusion for our study. Firstly, the highest frequencies of callus induction were observed on MS medium, supplemented with BAP and NAA hormones, together with optimized growth parameters. Second, SM analysis showed embryogenic characteristic in cells of the yellow fiber calli by the presence of small and isogeometric cells, and ECMSN, which was the extracellular metric surface network on the callus cells. For the PCR analysis, only targeted SERP gene was successfully amplified and enabled to proceed to in silico analysis. However, the partial fragment of sequence hit on isoleucine transferase conserved the domain, not the SCRK domain. So in future, we have to optimize these genes, particularly the PCR cycle, and we will study the full length gene isolation towards biomarker and functional genomic study. So these are the references for our study, and we want to acknowledge the financial support provided by University of Malaysia Pahang. So that's all from me. Thank you for your time and wish you all the best in this second international conference on tropical resources and sustainable sciences. Thank you very much and see you again.